So this is the EGFR pathway and proteins, and we're going to go through this today. Me, Hunter Panyer, and my group mate, Ryan Assini.
All right. So to start off this presentation, we're going to just briefly go over the generalized pathway for how EGFR works, and then we're going to dive into each of the different proteins that we mention and how they function, and specifically how their structure relates to their function. So overall, the EGFR is a pathway that can be activated by extracellular signalers, which can bind to an EGFR protein and lead to dimerization. The EGFR receptor itself can either form a homodimer with another EGFR protein, or it can form a heterodimer with another EGFR family protein. And these vary, but none of them are shown in this figure. This dimerization is what winds up activating the EGFR kinase pathway. There's a couple of different ones. There's the MAPK/ERK pathway, which is shown in blue, the AKT one, which is shown in orange on the right, and the JNK one, which is shown in green. We're going to be focusing on the MAPK/ERK pathway. And really interesting as a side note, with EGFR is overactivity or overexpression of EGFR is a characteristic of a lot of cancer cells. So treatments can target these proteins and try to lower some of their expression, which is what some cancer therapies do. All right. So the first protein that we're going to be looking at is EGFR itself. And EGFR as a protein is, it normally shows up as a monomer when it's in its unbound form. And this monomer, if you isolate one of these sides, the extracellular section is broken up into 4 domains. And when a ligand shows up, domains 1 and 3 wind up binding to the ligand, as shown over here, and domain 2 winds up binding to the other protein in the homodimer. The makeups of these domains vary. So the domains 1 and 3 are lysine rich, and domains 2 and 4 are cysteine and serine rich, and they alternate back and forth. So just continuing the EGFR structure, it has quite a few very specific structures that relate to its function. So on its N-terminal, it has the receptor binding domain, which is what we just talked about. Domains 1 and 3 bind to a ligand and change its shape. It is a transmembrane domain, which goes through its lipid bilayer, and that is the alpha helix that you can see on the right of this figure. It also has a couple of kinases with catalytic activity, and those can bind ATP and take part in the final process of the EGFR, which on its C-terminal tail, it's very rich in tyrosines, and this allows it to become phosphorylated when these proteins dimerize to each other. And that phosphorylation is important for the activation of the next protein we'll talk about, which is GRB2. GRB2 is an adapter protein, which is the second step of the MAP-ERK pathway. It has 1 SH2 and 2 SH3 domains, which are just very well conserved domains in proteins and are able to bind to a lot of different sites. What's important with these is that the SH2 domain binds to the phosphorylated TYR residues that are at the bottom of the C-terminal tail of the EGFR. So like we just talked about, the tyrosine amino acids get phosphorylated, and then they can then activate the SH2 domains in the GRB2. And Hunter is going to talk about the next two proteins.
So the next protein to look at is the RAS protein. So this is the next activation after the GRB2 binds to the EGFR. So RAS is normally bound to a guanosine diphosphate, and this can be shown here on the right. This is its inactivated form. So if we see here, it's actually pretty clear that we can see it's a very closed up structure here. So you can see the difference between the GDP and the GTP, which is the activated form. You can see that in the GTP form, we have this activation, or we have this opening up of the active site that allows it to then continue the chain, and continue the phosphorylation cascade. So if we move on to the next slide, we're going to then show the kind of a cartoon image of the GDP GTP activation. So it's kind of shown here as almost a spring-loaded activation. So this phosphorylation ends up making this GDP a GTP, and then these kind of switch proteins open up, and this opens up the active site. So it really kind of solidifies how the structure of this changes as it's phosphorylated. So now moving on to the next protein in the structure, or the next complex in the structure, is actually the RAS protein that we just took a look at, binding to the following proteins, the RAF, the MEK, and the ERK. And this cascade, the cascade that starts off with EGFR, moves along this RAS, RAF, MEK, ERK structure, and they subsequently phosphorylate each other. And this leads to the downstream activation. So we can see in the top structure, or the top part of this structure over to the right, the inactive versus the active forms of this large complex. And you can see the clear conformational change where the inactive form kind of has a closed off structure, and then the active form has this opening up top, allowing it to then proceed through the cascade. And then down below, we can see the binding and the eventual release of the p-ERK. So this structure kind of goes along, and then this complex down here is released because there's a lot of these proteins like RAS and RAF are membrane bound. So this phosphorylation then leads to the release of p-ERK, which can then go and activate transcription factors, or carry the cell signal, or carry the surface signal that starts with EGFR down to the rest of the cell, and lead to transcription factors activating genes or turning off gene transcription. And then we can move on to the next slide, which is discussing AP1 activation. So in this particular pathway that we're looking at, the signaling cascade leads to transcriptional activation of AP1. And if we click forward, we can see that the Jun-Phos two structures come together in this kind of X pattern, and this leads to, this activates its ability to bind to the DNA, and specifically that AP1 promoter region. So you can see on the image on the right, it's that X pattern, and then within the bottom of that X pattern is where the actual DNA segment is allowed to bind. And this is a dimerization of these two proteins, these two alpha helical proteins. And then moving on to the next slide, just brings us back to the overall pathway. just to kind of bring us back and show the overall view. So we started with the EGFR signaling, and it received an extracellular signal, which then used the EGFR activation to travel through RAS, RAF, MEK, and ERK all the way down to activate transcription of AP1. So this, as you can see here, there are three different cascades listed, but there are many that use this type of surface signaling through the membrane to downstream activation.
Okay. And I think that brings our presentation of the EGFR activated pathway to a close. If you have any questions, please post them in the discussion board. And this is a list of our references.